Well, thank you everyone for making it this far. We heard a lot about the different types of samples and approaches to analyzing our SAXS data today. Um, uh, I wanted to talk more about some of the tools that were developed by the staff at our Beamline and just some hands-on showing you what you can do with your data. Few things I want to say is a lot of these tools are made for biological molecules, so things that we have a known structure for. I'm going to talk a little about that later. So one thing that our group is trying to steer away in terms of protein structures with SAXS is staying away from what McCall calls blobology. So like trying to use these like bead models or ab initio structures to fit your data. And well, that's useful in many cases, especially if you don't have an initial model to start with, like we saw with Dr. Hackett with nanodiscs, or if we have some lamellar packing, what we saw in people today, that's you kind of need to start with. You don't have something to start with, so you have to reform it. But what we want to take advantage of in BioSAXS is putting as many input parameters as we can to get information, and that's when SAXS, I think, is really powerful. And this is all, by the way, not from our Beamline, it's from the SAS bio data bank repository. So I want to just share kind of how I see SAXS or BioSAXS, and the analogy I like to use is that we can see a SAXS curve as kind of a silhouette, like a shadow of a cool structure that we want to study. So when we have a shadow, we don't have the, we have some input information, or we have some like constraint information, but it's not really a full picture of it yet. And we have to, we could use that shadow itself and make up everything that's in there and get something that's a little crude. But like I was saying, I think SAXS is really powerful when you start out with a model to fit into the data. So in this case, it's a PDB or model of the protein that you measured. And so my analogy here is kind of these movable action figures where you know what the shape of that action figure is, and you can move all the joints in a way you want that can fit into the shadow. And so that can potentially reveal the position of any domains or functional groups in your protein. And like I was saying before, it's best to do this type of analysis with something you know is monodispersed and you have knowledge of full sequencing, but again, we don't always have that option depending on the type of experiment you're doing. So yeah, if we have a starting model, we can learn more from the SAXS. So now I'm gonna stop sharing my PowerPoint, and I hope that the Zoom can keep up. I'm getting kind of scared of that now, but I just want to show a few examples of the tools that you can use. So first I'm going to talk about FoXS. So that is a web app that you can access, that you can basically fit a PDB to your SAXS data by adjusting a few parameters. So we have, let's see, okay, it looks like it's working well. All right, so here's the web page, and what I'm going to show you now is, let's say we have data that's not that hasn't gone through chromatography, and we might have different oligomerization states in there. So what I did here is I have AlphaFold models from the sequence that was here in this data bank, and we're going to see if we can get a good fit with a combination of those, because the molecular weights that you can calculate from this SAXS doesn't really match up with the supposed dimer of the system. So let's see if we can. Okay, here's the FoXS. I'm going to just show you how you input one model. So here's where you go into, you click that, and you I'll go to this folder, and you put in your PDB here. So I'm going to try just fitting the dimer. Oh, wait, sorry, the PDB goes there.
And just a comment, you can grab and from window to window. I'm not sure if the Windows can do that.
Yeah, but I don't like doing that if I have
Oh,
screen.
okay.
I think I tried that. At least on Windows.
Windows is where.
So I'm going to hit submit. This will just take a second. And you can see, well, it's fitting okay. We have 2.87 chi-square, but our C1 and C2, this is the factors that represent excluded volume and hydration layer density are kind of pushed to their maximum, at least with the C2, in order to get, a reasonable fit. And you can see the residuals is kind of fluctuating here in the low, like the really low Q anyway, below 0.1, which means something's wrong with the oligomerization state. So instead, what we're going to do is use multi-FoXS. So I'm going to click back. And then, so here I have a folder here that I called multi-FoXS for today, and we have the models for the dimer, monomer, and trimer that we're just going to zip together. And now let's just do that thing Michal likes to do. Just put that in there, good, okay. And then when you go to advanced options and click this one, fit models into multiple structures to see if there is multiple options. I'm going to hit submit. So this one's taking a bit. So yeah, individually you can see that the dimer was at that 2.87, but the other oligomers are even worse. But I click this one here. It says multi-state models by multi-FoXS. I remember Michal earlier was complaining about how that button isn't more obvious on the website. But here we go. Okay, now it's saying if we have 50% of the dimer and 50% of the monomer, we can get a fit, a chi-square that's 1.91. Now that's better. Generally, I like to think 2.0 or lower is reasonable, but now we can see that, okay, it's clear that in this sample there is both monomers and dimers present, and we can get a rough quantification of how much are in both ends. Sorry, when I said 50%, I was looking at this guy right here. That's 0.498, and this guy here. And so this is just an example with oligomers. Michal was showing an example with different conformation states. If you have different models like that, to see what fits with the data. Because yeah, if you have different states in there, which is we like to avoid, but can happen, especially in the high throughput or just non-chromatography samples. You might have different states, and this is a good way of approaching that. Okay, so yeah, we can talk more in the next session if we have specific questions about our tools. But now I'm going to move on to what if we want to go beyond just initial model fitting, and we want to manipulate our PDB or structure to fit into our data. So this is a tool that Michal and Scott are developing, or have developed, called BilboMD. It determines the best conformation throughout, reasonable spaces defined by AlphaFold error models to fit it to the SAXS. So here's another sample from the SAXS data bank, and we have SAXS here, and we can learn a lot about the structure from the paired distance distribution function, which is here, and we can see that it kind of elongates until it's like 170 angstroms there, and so you can tell just from the P of R that it's very elongated. This protein, the structure is here. Actually, I'll just open it here. And it's got these long tails that are coming out of there. And this is just the way AlphaFold gave it to you. But it's clear that, from the manuscript that this is from, that those tails are what's causing the elongation. But in the manuscript that this is from, they were kind of pointing up and down, like parallel to this way. But, is that something that we can test, that we can fit into the SAXS? So what we do is we incorporate the information from the AlphaFold PA or the PAE matrix, and that information is here, and we can see that the flexible regions here are very high error, which is why they're red. And so what BilboMD does is it takes those flexible regions and it allows them to explore different conformations to fit the SAXS. So let's just real quick show you, just to demonstrate. I'm going to go to, and I have FoXS links there because it's something I go to all the time, So let's just real quick. Don't worry about that.
No, you have to make it
So let's look at the fitting of the initial AlphaFold and see how bad that fit is. Yeah, so it's not fitting very well, as you can see, and it's likely due to those flexible regions. So now we'll go to BilboMD. Okay, so I hope that no user password information or anything is being shared. I just thought of that now, but let's just go with it. So you have to make an account, and then we can talk about that later, but it's really user-friendly, I think, at this point, and we have Scott and Nicole to thank for that. So I'm going to input our AlphaFold PDB. That would be here. This is directly from AlphaFold, and then the JSON, which has that error information, along with our experimental SAXS data. And I'm going to call this demo. And I guess I'll hit submit to work that doesn't need to. And yeah, this is going to take about an hour, so I think we can all just sit here for an hour. No, I did it earlier. So yeah, here it is completed. I feel like Julia Child, this is like now I have the turkey that's completely done. So now we can see that chi-squared that was once 20 is now fitting a lot better, and it did move around those flexible regions. So we have it fits it with different numbers of ensembles. So this is 1, 2, and so forth, and we are getting better fits with increasing number of ensembles. They're all pretty fun. This was a really good example, but this isn't going to be the only example I show. But we generally like to pick like the best fit with the lowest number of ensemble states. And you can download them here, but I have it here already because this is Julia Child. Okay, so here's the ensemble states one, it was kind of a little bit worse, but you can see here with the two ensemble states, they're overlaid. Let's make it white. Maybe that's better. And I'm gonna make that color a little bit more easy to distinguish. That indeed that the flexible regions that we're moving around, we're not going up and down, they're kind of moving towards the sides, and that's kind of what the SAXS is suggesting. And that was two ensemble states, here's one ensemble state, and that's even more like horizontal, I'll say. So yeah, this was kind of a cool way to manipulate your model to fit the SAXS and kind of get a good guess as to what's going on in solution. Yes?
So how come you decided to use like PAE instead of like PLDDT?
So I think, yeah, that this is stuff that I don't remember that as well, but you do just put in like all the information from the AlphaFold error plot in there.
I can comment on that. It's using both.
Okay.
So PLDT is just using defining the rigidity and flexibility, like the confidence interval. And PAE is defining the cluster of the regions.
So,
Yeah.
Josh, if you bring up that PAE matrix in
That
Chimera
was-
and click the other button
Yeah.
that says color by domain.
So yeah, it's using both. It's
Yeah.
calculating first which region are flexible and then calculate which region is supposed to be together or interactive modes. So you can see the complex, these species are interacting or not interacting. All that is part of that development.
So is it essentially using the PAE to take into account geometry, whereas the PLDDT would be more so like rigidity? Is that like a good way to think about it, or?
Yeah, well,
PAE matrix tells us which domains
are
are
together.
likely going to be related to one another in a three-dimensional space. So click the button that says color by.
PAE.
Yeah, that's
So,
what this is.
so when you see these off diagonal blue and yellow squares, it indicates that that region is very highly likely to be oriented correctly with respect to the region that's on the diagonal.
So this is dimer. And PAE matrix recognize that this domain and this monomer interact with this exactly in this region. If there will be no confidency, that will be white. That means they're not interacting
And
at
in
all.
the molecular dynamics, we would allow them to move independently. Since there's an off diagonal region there, we know that they're forming a pretty rigid dimer, and we should not fuck with that.
And the yellow parts or the white parts here tell you there is no fault. That's the PLDT number. But that's the beauty of the AlphaFold, to give you a lot of this information that we can just plug it in a SAXS.
Yeah, yeah, so what I was showing you here was kind of just our color coding to show you the regions of higher, we weren't only using one or the other, for the thing, so, anyway.
There was a question in the chat as well.
Oh, yes. Someone help me out?
Do you want me to, do you see it? Oh,
I
sorry.
don't know,
Can we override the max file size of 20 MB for the PAE? I don't
I
know
was
what
just
that
talking to
says.
Scott
Get
about
it?
that,
I'll do this.
yeah, we
I didn't
can.
realize these files were getting so big, so I just had
Oh,
artificially small.
don't answer, yeah.
You can hear me.
Yeah.
Whoever asked the
Yeah, yeah.
question,
On-
I hear
on
me
Zoom.
talking, maybe they.
You already have.
I'll
Zoom
fix
up
it,
there.
it'll get
In that
fixed.
top bar, there's a chat.
I didn't make the issue,
Oh.
right?
Yeah.
Yeah, that's why you forget. Next time I should do the issue.
Once
Zoom is not visible, oh,
Oh,
Now, especially with AlphaFold 3, the those files have extra matrices in them now, so that they balloon are huge. So, yeah, all fixed. Sorry.
Okay, great things to point out. I think 50 might, we can talk later. But okay, I'm gonna show one more example with BilboMD. And that is what if we want to, which is something that I've been trying to explore since my time here, is adjusting the flexibility of the model in the input parameters, or the constraints of your model. So here we have, this one is not just one protein, but a complex. And the data bank had their own fit, but I kind of looked at it myself and it wasn't that good of a fit. I wanted to see if I can improve it. And so I did the same thing as before. Let's go to that. I took the AlphaFold complex, I put it in here, and we got this result. Okay, we have a chi-squared of 2, 3, 1, and we look at our residuals here of the fitted. This is the original model. This is it after the minimization. And yeah, I mean, 2.31 is okay, but it could be improved. But what I am looking for is regions around here between 0.1 and 0.2 inverse angstroms where there's really high error there. And that what that region is representing is regions of high flexibility. And it's clear that the constraints that this program took at first weren't, good enough to explore that many confirmations. So we're going to instead not go to BilboMD Auto, but what's called here in the tab as BilboMD Classic. Well, actually, first we have to go. Well, first I'll just talk about this. So here you have to upload what's called the const.inp file. This is what's going to define what residues in your protein are going to be fixed, rigid, or mobile. And the long flexible regions are what we want to move around and explore fittings for. So we're going to go to this PAE Jiffy TM. Well, I think Jiffy is its own company, and that's why it's TM. Anyway, so here we can get that const inp file, and we're going to go to this one. So we're going to put our PDB and our JSON, and the defaults. And so here's a parameter that we define as the clustering weight. So as you can see written here, well, maybe not, it's really small, but when we decrease this value, we'll have fewer clusters. So fewer bodies in our protein are going to be rigid, or are going to be flexible. And moving it to the right will make it less rigid. So there will be more clusters of rigid bodies, which means the stuff in between them are going to be flexible. So just moving it forward will make it more flexible, I guess. It will. I think of it as because we're adjusting like the threshold from the AlphaFold error. It makes more residues flexible. So yeah. So what we're going to do is, well, if we hit submit now, we get this constraints file. Oh, I guess that takes longer than I thought. And you can see here, it defines which residues are fixed, which are rigid, and everything that's not defined here is flexible. And then the details of this are in a paper from 2018. which I can share with everyone later. I don't have the link now, but yeah, I'm gonna just refresh this. But we want to try to increase the flexibility and try to fit this again. So I'm gonna move this to 5, and when I hit submit, I guess this will take even longer, maybe. And maybe not. But now I have to sit here.
It's just a reading of the PA.
Yeah, you can see that there's more rigid regions, so more clusters, which means that it's kind of like more beads on a string. So it's going to be more flexible. And you can download this constraints file, which I already did. But now we can, with this new, again, like the default value for that, this clustering value here was 2, but I can download that for 5. So now, you know, this was what was removed from Classic to make it auto, but we're going to go back to the Classic. We're going to call this demo 2. I'm going to put our PDB here, and I'm going to put our new constraints file, which I labeled here earlier as 5. And then our SAXS here, and then now from the SAXS it determines the possible RGs that it's going to fit the structure into, and you can define how many confirmations per RG we'll do. Let's just do 200 because we're not going to look at these results anyway. But I hit submit, and here's cooking show magic. Yeah, here is now the improved model, and you can see that specifically once we hit 2 ensemble states, we get kind of a cleaner fit. Let's just hide those. And then when we look at the models here, I also
Cooking magic.
Yeah, so here is the first one with, here's the BilboMD structure before we increased that clustering weight. And then here is this is 1 ensemble state, and then here's 2 ensemble states after doing that. And we can see that actually it's these residues, I'll go back to that guy. These residues here actually, if these were initially not flexible, but when we allowed more clustering, they were allowed to move around, and we can see that actually the angle of this entire thing might actually be tighter than what we would have assumed otherwise. So that's one other thing that you can do is try to get a good fit with your protein structure. Now, you of course have to think what's reasonable and what's not with your system, and a computer's not going to always know that, and will try to fit stuff like ridiculously and unfold helices and things if you're not careful with the constraints. But I hope that I gave people here a general idea of how to use these tools. One other thing I wanted to say that I didn't was you can actually, if you read the paper about the constraints files, you can also manually adjust what residues you want fixed or rigid in the const file. But yeah, I hope I showed people that if you have input information, SAXS can be a lot more powerful. That's my main point. So thank you. If anyone has a question now.
So how small of changes in the secondary structure do you think that this will be, like that the SAXS can pick up and that the model will be able to affect?
secondary structure, like if you have different oligomers.
Different oligomers, or say you have like a helix moving a couple angstroms, like how many angstroms will the SAXS actually be able to pick up?
Angstroms, I actually don't, but what I showed you here is we were able to pick up, that long. How long is that?
Depending on the size
And the symmetry.
and the quality of data. You can really literally see, I have the slides that I never really showed this. You can see the haystack versus, no, it's dramatic change, but the movement of helix is huge, even if it's 1 or 2 angstrom, but the whole body is moving, so the overall structure changes. So you can move 1, 2 angstrom, you see it in the SAXS data. It's a very good example using FoXS, 2 models that you have in your mind, put it in a FoXS, not fitting, but just calculate the curves, and you see how good data needs to be to see this difference. But it's really depending on the size of the protein and quality of data.
Thank you.
Here's some chat
Oh, there was one question in chat during the presentation.
Remind me of that.
from Todd. Todd, are you still there?
I am. I'm still here.
Hi.
Hi, Todd.
Yeah.
Hi.
Yeah, okay. Would you like to ask?
So I think this was discussed before, but I'm and then maybe I missed it. But I think AlphaFold generates like 10 different models. So I guess my question was, any plans to have like a server that will just grab all of those models and fit it to the data to see which one, gives the best fit?
Well, I would start with the FoXS, which can very much take 10 models, and then usually when I look at all the AlphaFold options, one of them is maybe a little bit better. And I use that as the initial model for the BilboMD.
So you go to FoXS first before you go to BilboMD.
Yes.
Okay, thank you.
But I like the idea with Scott, we discussed that just if we have a super computer nurse, we just gonna produce for every sample from the user AlphaFold run and speed up directly to BilboMD and fitting on all 5 or 10 models. Why not? But you get so much output data that you will be so much overwhelmed with so much feeding that you may potentially lost your understanding.
So Javier, I think, has a question on Zoom.
Okay, somebody is raising their hand.
Hi, hello, thank you so much. My question is, can we generate constraint files for protein nucleic acids with the PAE Jiffy? I've tried it with an AlphaFold multimer prediction, but it wouldn't run. I'm not sure if there's a workaround.
It's supposed to run, so maybe send us the files and we can look at this. It should run.
Yeah.
Okay.
Yeah,
Thank you.
It's under development, still beta version.